collaborator and friend, Dr. Kent Mao. Dr. Mao is a radiation oncology physician scientist here at Dana-Farber and Brigham and an associate professor of radiation oncology at Harvard Med School. His independent lab at Dana-Farber, they work on the interface of cancer genomics, DNA damage repair, and translational oncology with a focus on genitourinary cancers, as you'll hear today. Kent completed his MD and PhD at the University of Chicago, where he worked in the lab of Dr. Phoebe Rice using X-ray crystallography to study mechanisms of DNA recombination. Then he came here to Boston to do an internship in medicine at Brigham, as well as a residency in radiation oncology at a joint Harvard radiation oncology program. Somehow found time during that residency to do a postdoctoral fellowship in the lab of Alan DeAndrea, studying DNA damage repair and tumor biology, as well as in therapy response. And some of his seminal work during that time involved identifying an association between ERCC2 mutations and cisplatin response in bladder cancer, and he continues to study this in his lab today. Now, Kent is a true physician scientist with an ongoing clinical practice caring for patients with GU cancers at the Lank Center for GU Oncology here, and he's also the co-director of the Bladder Cancer Center. Now, his observations in the lab have led to new approaches in the clinic as well as ongoing clinical trials. And if you don't believe me with regards to his excellence in the clinic, you can hear it straight from one of his patients, and I quote, this patient said, "I can't say enough about Dr. Mao. He's so bright and knowledgeable, as well as pleasant and a joy, and it's a joy to be his patient. I have complete trust in him." And to that end, Kent's work has been also published widely, including in Nature Genetics, Journal of Clinical Investigation, Cancer Discovery, just to name a few. He's a sought-after speaker and a leader in the bladder cancer research world. He's a PI now of 3 ongoing R01s, as well as multiple foundation grants, and his accomplishments have been widely recognized, including a number of awards, the Burroughs Wellcome Career Award for Medical Scientist, Prostate Cancer Foundation Young Investigator Award, and a recent election to the American Society of Clinical Investigation. So on a personal note, Kent is a fantastic colleague, always thoughtful, always pushing for excellence in the clinic and the lab. With that, I'm looking forward to Kent's lecture today. Dr. Kent Mao.
Thanks, Joe. That was really nice. Yeah, I'm happy to be here to talk today about some of the work in my lab. These are the disclosures I have. None of these are going to be directly relevant to any of the work that I'll discuss today. So as Joe mentioned, our lab is focused really on the interface between DNA repair, GU tumor biology, and radiation oncology. And so the majority of my talk today will focus on projects at sort of these different overlaps. So our cells are continually exposed to both endogenous and exogenous sources of DNA damage. These DNA damaging agents create a variety of DNA lesions that thus require different repair pathways to fix the lesions and restore genomic integrity. These DNA repair pathways then signal through a series of DNA damage response kinases that both help coordinate repair and signal to cell cycle and other critical processes. There are good examples in oncology of specific DNA repair defects being therapeutically vulnerable. So great examples are homologous recombination deficiency, which can be targeted with PARP inhibitors in different clinical contexts, as well as mismatch repair deficiency, which can be targeted by pembrolizumab. Similarly, many of these DNA damage response kinases have small molecule inhibitors that are at different stages of clinical development, again, in an effort to really target these processes in tumors. I'm going to spend the majority of my time today talking about nucleotide excision repair, which is a relatively less well-characterized pathway in the cancer context. And so briefly, the NER pathway is a highly conserved DNA repair pathway that has evolved to repair these bulky intrastrand DNA adducts, like those caused by platinum drugs and UV sensitivity. NER can actually be activated via one of 2 subpathways. So the global genome, or GGNER subpathway, can detect lesions anywhere in the genome and activate the pathway, whereas transcription-coupled repair relies on transcribing RNA polymerase to encounter the lesion and then activate the pathway. These pathways then converge on a shared pathway that involves DNA duplex unwinding by DNA helicases, DNA excision by endonucleases to release a single-stranded portion of DNA that contains the lesion. A polymerase then comes in and fills in that gap, and that is the backbone is sealed by a ligase, such that you ultimately get error-free repair of the lesion. Two specific proteins that I wanted to highlight that I'll talk in more detail about today are, one is DDB2 or XPE. This is the protein that directly binds to the lesion to activate the GG-NER subpathway. And the other is ERCC2 or XPD, which is one of the DNA helicases that unwinds the duplex near the site of damage. Most of the genetics and the biochemistry of the NER pathway over the past decades have actually been elucidated through study of one of several inherited NER diseases. So the one I'm going to focus most on is xeroderma pigmentosum, or XP. So these patients have biallelic germline inactivating mutations in an NER gene. It's relatively rare, about 1 in a million live births in US and Western Europe, but with it comes a dramatic increased risk of UV-induced cancers of the skin, lips, tongue, etc. But as I alluded to, the role of somatic NER gene alterations is less well studied. And so to briefly shift, but again, by way of background, I wanted to introduce a few things about bladder cancer, which is the disease I mostly study and treat. So bladder cancer is, I think, more common than many people realize in the US. There's something like 80,000 new cases and 15,000 deaths each year in the US. Bladder cancer is staged based on the depth of invasion of the tumor into the wall of the bladder. And so the tumors all start on the inner epithelial lining of the bladder. Some remain superficial, and these are the so-called non-muscle invasive bladder cancers. However, a subset of the more aggressive tumors will actually invade into the muscle wall of the bladder to create muscle invasive bladder cancer, or MIBC. And then if left untreated, the disease can spread to the nodes or beyond, and that sort of rounds out the staging criteria. Thankfully, the majority of the patients at diagnosis have non-muscle invasive disease, and this is a disease that's typically managed primarily by urologists with cystoscopic resections and intravesicular therapy. But I'll be focusing the majority of my talk on about a quarter of patients who are diagnosed with either muscle invasive or more advanced bladder cancer. And as you can see at the table at the bottom, prognosis is strongly linked to stage of diagnosis. So most patients with non-muscle invasive bladder cancer do well, whereas the prognosis for muscle invasive or metastatic patients is much more guarded. So cisplatin has a long history of use in both muscle invasive and metastatic bladder cancer. In the muscle invasive space, there's good randomized trial evidence showing that the addition of neoadjuvant cisplatin-based chemotherapy improves survival compared to cystectomy alone. Cystectomy is bladder removal surgery. And so by far the most common curative intent treatment option for MIBC in the US is neoadjuvant chemotherapy followed by radical cystectomy. And studies of these patient populations have demonstrated that the survival is closely related to the response of the disease at the time of surgery. So that is to say, patients who have a complete response, meaning the tumor is completely gone after chemotherapy at the time of surgery, they have a really good prognosis. Whereas patients with partial response, meaning that their tumor shrinks from being muscle invasive to non-muscle invasive, also have a reasonably good prognosis. But it's that subset of patients with stable or progressive disease, despite chemotherapy, that have unfortunately the worst prognosis. However, historically, it's been difficult when you meet patients to determine who will versus won't respond to this platinum-based chemotherapy. And so this was sort of the motivation for one of the first studies I embarked on as a postdoc here with other investigators at Farber at the time. And so in this study, we accrued 50 muscle-invasive bladder cancer patients, all of whom got neoadjuvant cisplatin-based chemotherapy and then had their bladders removed. But we selected the cohort to include 25 patients that we called responders. These are patients who had complete response and no tumor at the time of surgery, and then 25 non-responders, meaning 25 patients who had residual muscle-invasive disease at the time of surgery. And we did exome sequencing of tumor and matched normal. And so this is an overview of the mutational landscape of the cohort. The responders are under the black bars on the left, the non-responders under the yellow bars on the right. And you can see that it's sort of the usual suspects in terms of the most commonly mutated genes across the cohort. Genes like P53 and RB1 have really well-described roles in bladder cancer biology. And you can sort of appreciate that the frequency of mutations in these genes is roughly similar between responders and non-responders. And there was only one gene, ERCC2, that was significantly enriched in responders relative to non-responders. There were 9 of 25 responders that had a somatic missense mutation in ERCC2 versus 0 of 25 non-responders. This is the same data shown in a different way. So on the x-axis is the mutational significance across the entire cohort of 50 patients, whereas on the y-axis is enrichment of mutations in responders relative to non-responders. And you can see how ERCC2 sticks out in that regard. So we've demonstrated this correlation in multiple cohorts now. And we then, if you follow the patients long enough, can track their survival. And what we found is that the improvement in pathologic response at the time of surgery translated to improved overall survival. This is in our initial discovery cohort as well as in a separate validation cohort. So ERC2 mutant patients generally do better than the wild type patients when treated using this paradigm. And so this is the paradigm that I just described, neoadjuvant chemotherapy followed by radical cystectomy. There's, however, another curative intent treatment option for these muscle-invasive bladder cancer patients, and this is actually most of my clinical practice, is so-called bladder-sparing trimodality therapy, where instead of having their bladder removed, the patients get a tumor resection followed by concurrent chemoradiotherapy. And the chemo that's given most commonly in this paradigm is cisplatin. And so we wondered whether patients with ERCC2 mutations may actually have better outcomes with this cisplatin-based chemoradiotherapy. And so this is a cohort of patients accrued with my colleagues at MGH, and we saw the same association where patients with ERCC2 mutant tumors had better bladder intact event-free survival than patients with a wild-type ERCC2. And so given these clinical observations, we've been motivated over a number of years to functionally analyze ERCC2 and its role in bladder cancer. And so to start out, we began with a relatively straightforward complementation assay where we took an ERCC2 deficient cell line and then added back either wild type or mutant ERCC2. And so you can see that when you add back the wild type version of the enzyme, you partially rescue the cisplatin sensitivity. However, when you add back one of the mutants that was observed in the cisplatin responsive clinical patients, you fail to rescue the cisplatin sensitivity, suggesting that these mutations are conferring inability to repair cisplatin-mediated DNA damage. We saw similar findings with UV. So UV creates DNA damage that's typically repaired by NER. You add back wild-type ERCC2 to deficient line, you rescue UV sensitivity, whereas if you add back one of the mutants, you fail to rescue sensitivity. We've done some limited in vitro biochemistry work. So this is with purified full-length or wild-type or mutant ERCC2. This is a radio-labeled ATP on a thin layer chromatography assay. And so you can see the wild-type ERCC2 relatively robustly hydrolyzes the radio-labeled ATP to inorganic phosphate, whereas a known ATPase mutant or one of these three different clinically observed ERCC2 mutations has a lot less ATPase activity. And because ERCC2 is a helicase, the ATPase activity and the helicase activity are tightly linked. And so this again provided some more evidence that these mutations that we were observing clinically were hampering ERCC2's normal function. Despite this biochemical evidence and testing cisplatin sensitivity, we've also been motivated to develop assays that directly measure nucleotide excision repair in cells. And so this is an assay that was developed by Jean-Bernard Lazaro, who's now a senior scientist in the lab. The way the assay works is that you plate the cells and then UV radiate them, incubate them for different lengths of time, and then fix the cells and add what we call a DDB2 proteoprobe. This is a protein purified from HeLa cells. And if you remember, the DDB2 is the protein that binds tightly and specifically directly to the UV-induced damage. And so we purify it from the cells, and we use it like you'd use an antibody in an immunofluorescence assay. And so 5 minutes after damage, all cells have lots of foci because they all have lots of damage. If you wait a couple hours and then fix the cells that have an intact NER pathway will have repaired the damage, and therefore you don't detect any remaining residual damage foci. Whereas in an NER deficient line, you have these residual foci indicative of unrepaired damage. And you can quantify these differences using automated microscopy. So this is what the data looks like, and we've reported this in a variety of different ways. So prior to damage, there's no foci. 5 minutes after damage, all of the cells have foci. And then in the wild-type ERCC2 complemented line, you get repair of the damage after 2.5 hours. whereas cell lines complemented with one of these two ERCC2 mutations identified from platinum-responsive patients, you fail to rescue the NER defect, suggesting, again, that these ERCC2 mutations are unable to support normal cellular NER. And so as the number of cohorts of bladder cancer patients sort of that are sequenced continues to expand, there's this sort of growing list of ERCC2 mutations. And we've been motivated to try to develop assays that allow us in a relatively high throughput way to measure the impact of these mutations on ERCC2 function. And so this is an assay that we've begun using more recently. It was developed by our collaborators Klaus Sorensen at the University of Copenhagen. And the way this assay works is that you transfect cells with Cas9 as well as an sgRNA that targets your mutation site of interest. You also transfect equal ratios of single-stranded donor templates, one of which contains the point mutation of interest, the other of which introduces a silent mutation that doesn't disrupt the normal reading frame, but allows you to track repair events over time. And then you can propagate these cells either with or without treatment to understand the impact of the mutation on proliferation and on drug sensitivity. And so just as sort of examples of what the data look like, so we typically normalize the mutant to wild type ratio at day 2. So that's the blue bars that are at the ratio of 1. If a mutation that you introduce doesn't have an effect on proliferation or sensitivity, then you expect that ratio of 1 to be maintained over time. If the mutation impacts proliferation at baseline, then you expect the green bar, the day 12 representation of the mutant repair event to go down. If the mutation doesn't impact baseline proliferation but does impact drug sensitivity, then like in sample C, you expect to see a decrease in the day 12 population in the treatment condition. And so we've done this now as proof of principle for a series of ERCC2 mutants, and the patterns are pretty clear. So none of these mutations impact baseline proliferation. So the green bars are all not significantly different than 100% here. Whereas the mutations, when you introduce a mutation in one of the two helicase domains of the protein, you dramatically increase sensitivity to cisplatin. Whereas when you introduce these mutations in a non-helicase domain location, you don't impact cisplatin sensitivity. One of the interesting features of the ERCC2 mutations that we observe clinically is that they're present as heterozygous mutations, meaning that they impact one allele, but the other allele does not undergo LOH, which is different than most DNA repair gene deficient settings where you need to inactivate both alleles, either by mutation or by deletion, in order to exert the phenotype. And so most of the functional work I've shown up to now hasn't truly modeled the heterozygous state that we think the tumors are in. And so we were interested in adapting this CRISPR select assay specifically to analyze that heterozygous state. And so using the sort of off-the-shelf assay, which is what I showed on the last slide, you have the cut at the mutation site of interest, and then you can get creation of the mutation of interest on one allele. However, these repair-directed events are still relatively uncommon, and so the single most likely event on the non-mutant allele is to create a Cas9 directed break followed by repair that generates an indel in that exon, and that's reflected in the pie chart here. The vast majority of events on the non-mutant allele are indel events in the exon, most of which lead to nonsense-mediated decay. And so what you're really modeling is a hemizygous disease state where you have the presence of the mutant enzyme but no wild-type enzyme. And so we modified this assay and designed the sgRNAs to cut within the intron that's adjacent to the exon that we wanted to modify. This way, you can still get repair and creation of the mutation of interest, but then when you have an indel on the opposite, on the other allele, it occurs within an intron and is less likely to disrupt the open reading frame. And that's again what we see here. Nearly all of the indel events are happening in the intron, and that allows you to then more accurately model the heterozygous disease state where you have one copy of the mutant enzyme and one copy of the wild type enzyme. We've compared results from these two approaches for a couple of the most common ERCC2 mutations, this N238S mutation and the D609G mutation. And what you can see is that whether you have the exon guide targeted sgRNAs, which generate this hemizygous disease state, or the intron guided, which is the heterozygous, both of them pretty potently sensitized to cisplatin. And so this is some preliminary evidence, and these are ongoing studies, to us that having the presence of a mutant ERC2 in the heterozygous state is sufficient to drive cisplatin sensitivity. And so putting it all together, this is the position of some of the most common mutant alleles mapped on the crystal structure of ERCC2. You can appreciate that many of the mutations sort of line up near this interface between conserved helicase domains, and mutations at those positions typically wipe out function as defined by cisplatin sensitivity or our NER assays, whereas the less common mutations that occur on the surface of the protein generally don't impact function. And then finally, we've been motivated to study this in vivo as well. And so to do this, we took a bladder cancer cell line that had wild type ERCC2, and we used CRISPR to introduce a mutation in the ERCC2 gene, and then put these tumor lines back into mice. When you don't treat the mice, the tumors grow at roughly the same rate. But when you treat the mice with cisplatin, it's really only the ERCC2 mutant tumor model that significantly responds. And so this was suggested to us that the mutation was sufficient to drive platinum sensitivity in vivo in this preclinical model. So at the same time that we had been studying ERCC2 in some detail, we and others were also interested in the impact of mutations in other DNA repair genes on cisplatin sensitivity in bladder cancer. And so these are some studies that have been published in this space showing a similar association, meaning that patients who have a DNA repair gene mutation in their tumor generally have better outcomes following cisplatin-based chemotherapy than patients who lack a mutation in one of those DNA repair genes. One of the other genes that we were most interested in studying was ATM. ATM is a DNA damage response kinase that's activated following DNA double-stranded breaks. ATM activation is required to help orchestrate the repair events at the breaks, as well as signal through a kinase cascade to do things like pause the cell cycle while the DNA is repaired. ATM was of interest for us because, in part, ATM mutations are relatively common across solid tumor types. This is data from the Bladder TCGA. You can see that the overall mutation rate is about 10%. Most of those mutations are missense mutations with lower percentages of truncating or splice site mutations. And so we modeled this ATM loss in our bladder cancer preclinical systems across a variety of the models. I'm just showing some here as representation. If we knock out ATM from bladder cancer cell lines, you abrogate normal DNA damage signaling, here shown by lack of radiation-induced gamma H2X foci in the knockout cell lines compared to the wild-type cell line. Similarly, the knockout cell lines have pretty significant increased sensitivity to DNA damaging agents like radiation and cisplatin. I'm not going to talk about it more here, but we also saw increased sensitivity to other DNA damage kinase inhibitors like ATR. One of the challenges with ATM, as I showed a couple of slides ago, is that ATM is a big gene. Many of the missense mutations that occur, it's difficult to predict their impact on function and on protein stability. And so we were motivated to develop an ATM IHC assay as a sort of corollary way to assess ATM function in tumors. And so working with our pathology colleagues, we optimized an IHC approach, and you can see the difference in staining patterns in tumors that have lost ATM versus in which the ATM staining is retained. And we used this to stain a cohort of tumors from patients treated with curative intent chemoradiotherapy. And what I'm showing here is we took all of the tumors that had any alteration in ATM as defined by NGS, and we applied the ATM IHC assay. And what you can see here is that the tumors that have ATM loss, meaning no functional or no full-length ATM protein in the tumor, had significantly better outcomes in terms of bladder intact event-free survival than in patients that had retained ATM, suggestive of more wild-type-like ATM function. And so, given the data that existed for different DNA repair gene alterations and platinum response in bladder cancer, there are a couple of clinical trials that are now open. So this, I'm highlighting one. This is run through the Alliance Cooperative Group. So patients with muscle-invasive bladder cancer are enrolled and undergo tumor sequencing. All patients then receive standard of care cisplatin plus gemcitabine neoadjuvant chemotherapy. At the end of chemotherapy, all the patients get clinically restaged by cystoscopy and by imaging. And if the patients have a complete clinical response and they have a mutation in one of these DNA repair genes, then rather than going to cystectomy, which would be the standard of care, they're allowed to undergo a close surveillance protocol in an effort to help them keep their bladder. Whereas if the patients either don't have a clinical response or don't have a mutation in one of these genes, then they get standard of care radical cystectomy. And there's a couple of these types of trials with similar designs that are now, some are ongoing, some are beginning to report. And so to sum up sort of the first part of what I've talked about here, somatic ERCC2 missense mutations are present in about 10% of bladder tumors. We believe that these heterozygous helicase domain ERCC2 mutations are sufficient to knock out NER function and sensitize cells to cisplatin. There are mutations in other non-NER DNA repair genes that also occur in bladder tumors and are associated with increased sensitivity. And these preclinical associations are being tested in multiple ongoing phase 2 trials to determine whether ERCC2 or these other DNA repair alterations can identify a subset of MIBC patients who may be cured with chemotherapy alone. So I wanted to turn now, most of the things I've talked about thus far have been about cisplatin sensitivity in bladder cancer. But only about 50% of bladder cancer patients are actually eligible for cisplatin-based chemotherapy due to the presence of a medical comorbidity like renal insufficiency or neuropathy. In addition, when patients develop tumors that are resistant to cisplatin, the prognosis is very poor. And so given this association between NER deficiency and cisplatin sensitivity, we were interested to understand whether there may be other agents that also could specifically target NER deficiency. And that's how we came across irofulven. Irofulven is a semi-synthetic derivative of a fungal product called aludin-S, the structure is shown here. Irofulven is a DNA alkylating agent, but it's unique in that it creates damage that doesn't disrupt the DNA backbone or the major groove architecture, and thus is more or less invisible to many DNA repair pathways, including base excision repair, mismatch repair, and global genome NER. Instead, the irofulven-mediated damage relies on transcribing RNA polymerase to run into the lesion and activate TCNER. And so if you remember, TCNER was one of the subpathways of the NER that feeds into this common NER pathway. And it's actually been shown for many years that in non-cancer systems, loss of function of a TCNER specific gene or a gene in the common NER pathway was sufficient to sensitize to irofulven-mediated DNA damage. And irofulven itself was actually tested in a series of phase 1 and 2 trials in the early to mid-2000s. And I'm just pulling a quote from one of the abstracts here. It said, irofulven administered at this dose and schedule was well tolerated, but had modest activity as a single agent. And these were, of course, non-biomarker-driven trials. And so given our observation that there were a subset of NER-deficient bladder tumors, we were interested in understanding whether they may be sensitive to irofulven. And so we tested irofulven in a series of our isogenic models, and we saw pretty significant increased sensitivity in the NER-deficient compared to the NER-proficient cell lines. And then in vivo as well, there was a dose-dependent tumor response in an NER-deficient xenograft model, whereas an NER-proficient model had no response at the same dose. And because it's an orphan drug, using it in trials in the US is a little complicated, but we have collaborators in Denmark who have a phase 2 trial of irofulven in NER-deficient metastatic bladder cancer that's planned to open later this year to test this hypothesis that NER deficiency may sensitize to irofulven. So I've talked mostly about bladder cancer so far, but a natural question is, are there NER gene alterations in other tumor types? And this is data from Memorial Sloan Kettering, their IMPACT data set. And you can see that small but real percentage of patients across solid tumor types have a potentially deleterious NER gene alteration in either the germline or their tumor. Alex Neal is a molecular pathology fellow here who's taken a deep look at our Dana-Farber OncoPanel data, which actually has better representation across NER genes than IMPACT does. And here you can see sort of the distribution of colors represents the different NER genes and their mutational frequency across solid tumor types. And so you can see some patterns, sort of common patterns emerge. So in bladder cancer, which is the third bar over, you see this big green here, which represents all the ERC2 mutant tumors in bladder cancer. And we won't have time to talk about it much today, but our group and others have, at least in the preclinical space, demonstrated that, yes, there are models that are NER deficient across a number of different tumor types. And these NER deficient models are more sensitive, as one would expect, to drugs like cisplatin and irofulven. So another way to try to identify NER deficiency, besides simply looking at NER genes and their mutational status, would be to look at mutational signatures. There's good data from other DNA repair deficient settings that things like mismatch repair deficiency or homologous recombination deficiency generate very characteristic patterns of mutations in tumors. And so we were interested in understanding whether that may be the case in NER deficiency. And so this is some older work that we did showing that ERCC2 mutations are very strongly correlated with a unique single base substitution signature in bladder cancer. This is data from across a composite cohort, but we've validated this in several individual cohorts as well. But perhaps more interestingly, we asked whether this signature activity was related to cisplatin response. And so that seemed to be the case. And so what I'm showing here is the activity of the signature on the y-axis in patients that have demonstrated to respond to platinum-based chemotherapy versus not. And you can see that even when you ignore ERCC2 status, there was more activity of the signature in the responders compared to the non-responders. We've continued to try to refine these mutational signature approaches. This is now a similar effort, but now using a composite signature that not only takes into account single base substitutions, but also things like insertions and deletions and transcription strand bias. And again, you see a very strong correlation between the strength of this composite signature and the ERC2 mutations, which are the green bars. Again, we were most interested in correlating this with something clinically meaningful. And so the bottom here is showing the signature activity by cisplatin response. And so when you consider all cases together, the orange bars are the cisplatin responders, the black bars are the non-responders. You can see that the signature activity, which is on the y-axis, is generally higher in responders than non-responders. But the caveat is that many of these responders had ERCC2 mutations. And so you could argue, well, you thought those were going to respond anyway because of the data I'd shown. And so even when you remove those cases, there still was an association, albeit less strong, between the signature and likelihood of cisplatin response. And so we're sort of continuing to press on this to understand whether there may be mutation-based ways from exome or genomes to infer NER pathway activity. And so to summarize those few parts, so irofulven is a small molecule that specifically targets NER deficient cancer cells, including cisplatin-resistant models, which I didn't really have time to show. And there's a clinical trial of irofulven in the metastatic NER deficient bladder cancer space that's planned. NER gene alterations are present across tumor types, I would say, albeit at a lower frequency than bladder cancer generally, and with unclear, as of yet at least, clinical implications. And then finally, ERC2 mutations in bladder cancer are associated with unique mutational patterns or signatures that also appear to correlate with cisplatin response, independent of ERC2 mutation status, although certainly additional work is needed. And so in the last couple of minutes here, I'd like to talk about some of our work in new areas of therapeutic advance in bladder cancer. One of them is antibody-drug conjugates, and one is targeting tumor-immune interactions. So I think people are probably aware, but antibody drug conjugates, they're really transforming the treatment landscape of bladder cancer. They're composed of an antibody that targets a cell surface protein on tumor cells, and then they're linked to a cytotoxic payload via a linker. In bladder cancer, there are numerous of these ADCs that are under clinical development. I'll talk about just the top two here today, because those are the most clinically advanced. So enfortumab vedotin is a Nectin-4 targeting antibody that's linked to MMAE, which is a microtubule inhibitor. And then sacituzumab govitecan, or SG, combines a TROP2 targeting antibody with govitecan, which is a type 1 topoisomerase inhibitor. And this is clinical data from the EV302 trial that was published just a little over a year ago, showing the activity of these ADCs in the metastatic disease space. And so here you see a significant improvement in overall survival in patients receiving the combination of enfortumab vedotin plus pembrolizumab compared to cisplatin-based chemotherapy. And this really is the first advance in this first-line disease setting in a couple of decades. And so lots of excitement around ADCs in bladder cancer. And so given my lab's interest in radiation and my clinical interest in using radiation to treat bladder cancer, we were interested in understanding and characterizing the interactions between ADCs and radiation in our preclinical bladder cancer models. And so we first asked, how does radiation impact expression of the targets in bladder cancer cells? And I would say, I'm showing representative data here, but I would say overall the effects were mild, but there were instances where radiation in both a time and dose-dependent fashion tended to increase expression of Nectin-4, which is a target of EV, and TROP2, which is a target of SG. And you can see that here as well on the immunofluorescence. You see increased membranous expression of TROP2 following radiation in the cell line. We next asked, how do these combine, what kind of combined cytotoxicity activity do they have? And so in general, across a variety of different preclinical models, we saw mostly, I would say, additivity. There were some settings in which we had a little bit of synergy. And then on the bottom here, I'm showing how we often think about radiation sensitizer behavior. And so the black bars are the survival curves with radiation alone, and then the red bars are when you add a dose of the payload of either EV or SG. You can see that they increase sensitivity of the cells. And then finally, we moved in vivo. And to do this, we took advantage of the Dana-Farber small animal radiation research platform, which allows us to deliver image-guided fractionated radiation. And so we implanted tumors orthotopically in mice. They're luciferase tagged, so you can track tumor growth by BLI. And then we treated the mice with ADC alone, radiation alone, or the combination. And for both EV and SG, you can see in the tumor growth curves here, better tumor control with the combination than you see with either agent alone. And these were treatments that were generally well tolerated, as shown by the mouse weights were relatively stable over the course of the experiments here. And so, given the general excitement about ADCs in bladder cancer, and given some of this promising preclinical data, there are now a couple of trials underway combining radiation with ADCs in the muscle-invasive disease space. Given our interest in the role of DNA repair deficiency and sensitivity to different agents in bladder cancer, it seemed natural to ask how different DNA repair pathway alterations impacted sensitivity specifically to the payloads from these newly approved ADCs. And so here, I'm showing an example of an HR-deficient bladder cancer model. Here we depleted BRCA2 to generate HR deficiency. And as expected, that leads to pretty significant increased sensitivity to both cisplatin and the PARP inhibitor olaparib. In addition, we saw increased sensitivity to MMAE, which is the payload of EV, as well as SN38, which is the payload of SG. Conversely, in our NER-deficient cell line systems, we did see increased sensitivity to cisplatin, as I shown before, but we didn't see increased sensitivity to either MMAE or SN38, suggesting, at least to us, that even though these ADCs are becoming approved in the sort of the first-line biomarker unselected patient population, there may be subsets of patients such as those with NER-deficient tumors, that may still benefit more from the old standard of care of cisplatin compared to either one of these new ADCs. And then finally, I wanted to talk about some of our work studying tumor microenvironmental factors. And so up to this point, most of the tumor alterations I talked about were tumor cell intrinsic drivers of cisplatin sensitivity, so ERCC2 mutation, ATM mutation, et cetera. But I think there's increasing awareness that the microenvironment can confer resistance to therapy. And so we were specifically interested in studying that in a bladder cancer setting. And so to do that, we actually undertook a study that in many ways resembled the initial study that I described for ERCC2, in that these are muscle-invasive bladder cancer patients, all treated with cisplatin-based chemotherapy, and then all having their bladders removed. And once again, we accrued patients who were either responders, in which case, at the time of surgery, there was only a tumor scar, or non-responders who had persistent muscle-invasive tumor at the time of surgery. And here we did single nucleus RNA sequencing and spatial transcriptomics, followed by some functional validation, and I'll show a little bit of that today. And so first we looked in the urothelial cell compartment, and unsurprisingly, given that there was persistent tumors in our non-responders and really just healing urothelium in the tumor scar, there was quite a bit of difference, as you can appreciate here. But our main interests were actually in the non-tumor cell compartments. And specifically, in this case, we noticed this interesting difference in myeloid cells. And so there was a significant enrichment in the non-responders, this population of tumor-associated macrophages characterized by high expression of SPP1 or osteopontin, which is a secreted factor that's been correlated with therapy resistance in a variety of different contexts, as well as PARP14. PARP14 is not the famous PARP. So PARP1 is sort of the PARP that's been implicated in DNA repair. It's the PARP inhibitor that's mostly targeted by approved PARP inhibitors. PARP14 is actually an immunomodulatory PARP. And so there was a big enrichment of this specific population of TAMs in non-responders compared to responders. And at the spatial transcriptomic level, you can appreciate maybe that there was co-localization of SPP1 expressing myeloid cells with CD44 expressing tumor cells. And that's represented in this yellow color. The yellow represents co-localization of these two cell types within 3 different tumors from non-responding patients. And so our hypothesis was that perhaps this specific subset of myeloid cells was helping drive cisplatin resistance in these non-responders. And so to do that, we've done a series of functional experiments. First, we simply took macrophages in vitro and treated them with increasing doses of cisplatin, and we observed increasing PARP14 expression on the macrophages as the cisplatin dose went up. We've also done some work using an immunocompetent mouse model. And so here, the mice are implanted, they develop tumors, and then they were treated with either PARP14 inhibitor alone, cisplatin alone, or the combination. And so you can see that the PARP14 inhibitor alone, which is commercially available, didn't have really much anti-tumor effect. The cisplatin in red has some anti-tumor effect, and then the combination does better than either of the monotherapies alone. And then at the end of this experiment, we harvested these tumors and we did RNA-seq. And what we saw was that there were several PARP14 target genes that were upregulated. And these are genes like CXCL11 and TLK7 that are known pro-inflammatory, anti-tumor. And so consistent with the idea that PARP14 inhibition is relieving this anti-tumor, promoting this anti-tumor effect through these cytokine networks. And that PARP14 inhibition may be a strategy to overcome cisplatin resistance in muscle-invasive bladder cancer. So to summarize the last part of the talk, ADCs are really transforming the treatment landscape for advanced bladder cancer. And we showed that they could be combined effectively with radiation in our preclinical models. HR and NER deficiency have differential impacts on sensitivity to the ADC payloads. our single cell work identified this population of part 14 SPP1 positive TAMs that were enriched in the cisplatin resistant MIBCs and co-localized with tumor cells at the spatial level. And that led to the hypothesis that we tested and showed that part 14 inhibition was able to increase cisplatin sensitivity in our immunocompetent mouse models. So yeah, I summarized, what I think is much of the work going on in the lab, but that we continue to stay busy. I think we're, particularly interested in continuing to identify and target NER deficiency both within and beyond bladder cancer. And so this could include attempts like new agents, so next generation irofulvins, direct inhibitors of NER proteins, and then new biomarkers, whether they be signature-based, which I touched on a little bit, or protein and IHC-based. I think we're really excited about the potential to target tumor TME interactions and other extrinsic determinants of DNA damage sensitivity in bladder cancer. One project, we're looking at targeting the CD44 SPP1 axis directly. And then at a sort of more basic mechanistic level, defining novel mechanisms of NER gene regulation. And so we've done a series of drug and genetic screens that have identified in some cases, new roles for known regulators of NER, and in some cases, new players altogether. And so I'm really excited about that work. It was a little, not quite ready for prime time today, but ongoing in the lab. And so, yeah, with that, I'd like to conclude and thank people, most specifically members of my lab, both current and former, who have done almost all the work that I showed today. I'd like to thank other members of my department. I think I have ongoing collaborations with essentially all of these people in my department. I'd like to thank my clinical colleagues in radiation oncology, medical oncology, urology. I collaborate closely with a number of different labs at Farber and beyond. I'd like to thank them. And then, yeah, certainly other folks beyond Boston as well. And then my funding sources are at the bottom. So thanks a lot for your attention. I'm happy to take questions.
Questions?